Hi guys, and in this video we are going to learn about unit number 18 heredity. In the topic is hereditary material. First of all, we want to learn the learning objectives of these videos. Just observe the topic map, and in this video we are going to learn about the two topic, that is genes and chromosome is one topic and DNA is another topic. According to the genes and chromosome topic, under we are learn about what are genes, what are chromosome, and structure of genes and chromosomes, regions, type and karyotyping. And in structure of DNA, what is DNA and structure, Watson and Crick model, and DNA replication and significance of the DNA. These following topics we are going to learn today. And first of all, genes and chromosome. What are genes? Genes are segment of the DNA which are responsible for the inheritance of a particular phenotypic character. And phenotypic character means external expression of a particular trait is known as phenotype. And a specific position of gene on a chromosome is called locus. It's a very important one. What is locus? Locus means a specific position of gene on a chromosome is called locus. And the function of gene means they pass on the genetic information from one generation to another. So it carry the hereditary information, that is genetic information, from one generation to next generation. Next, what are chromosomes? A thin thread-like structure present in the nucleus is called chromosome. A thin thread-like structure present in the nucleus is called chromosomes. The term chromosome was first coined by Wilder in the year 1888. And the chromosomes are carrier of genetic material from one generation to another generation. They contain the hereditary information. And you can see that structure of chromosomes, and this is the centromere, and this is chromatids, and this one is sister chromatids. Like that, this is also chromatid, this one is sister chromatid of the chromosomes. And next, structure of chromosome. First, sister chromatid means two identical strand of chromosome is called sister chromatid. And centromere, the connection between of the sister chromatids is called as centromere. And chromonema means a spirally coiled thin structure is called structure of chromatid is called chromonema. And chromomeres means a bird like structure, means bell, money mari irukra structure present along the chromonema of the length is called chromomeres. And structural composition of DNA and RNA. DNA and RNA chromosomal proteins, that is histone and non-histone, and certain metallic ions. It's very important. And this protein provide the structural support of the chromosomes. And next, regions, that is primary construction. Primary construction is nothing but it is a centromere is called as primary construction. The two arms of the chromosome meet at one point is called primary construction, which means the two arms of the chromosomes, that is this is one arm, this is two arms. So these two arms are meet at a point is called as primary construction or centromere. And the centromere is the region where the spindle fibers attach to the chromosomes during the cell divisions. So during the cell division, centromere is separated by the spindle fibers. And secondary construction, some chromosome possess a secondary construction at any point of the chromosomes. They are known as a nuclear zone or nucleolar organizer. So nuclear zone or nuclear organizer the formation of nucleolus in the nucleus. It's very, very important point. And next, telomeres. The end of the chromosome is called telomeres. That is, each extremity of the chromosome has a polarity and prevent it from joining and adjacent chromosomes. So, in the rendu chromosomes pathina onna seradu, and it maintain the provides stability of the chromosomes. And next, satellite. Some of the chromosomes have an elongated knob-like appendage at the end of the chromosomes as known as satellite. The chromosome which satellite are called as sat chromosomes. Chromosomes are classified based on the position of the centromere into 4 types. First, telocentric chromosome, acrocentric chromosome, submetacentric chromosome and metacentric chromosome. And telocentric chromosome means the centromere is found on the proximal end. There are rod shaped chromosome. And acrocentric chromosome means the centromere is found at the one end with short arm and another end is a long arm. So you can see the diagram also. And submetacentric chromosome means the centromere is found near the center of the chromosomes. Thus, the forming of 2 unequal arms there are shaped as L-shaped chromosomes. And metacentric chromosome means the centromere occurs in the center of the chromosomes and form 2 equal arms. There are V-shaped chromosomes. Telocentric chromosome is a rod shape. Acrocentric chromosomes also a rod shaped chromosome, and submetacentric chromosome is a J-shaped or L-shaped chromosomes. Metacentric chromosome is a V-shaped chromosomes. Based on the functions, chromosomes are classified into 2 types, that is autosomes and allosomes. Autosomes means body chromosomes, allosomes means sex chromosomes. Okay, autosomes. It contains the genes that determine a somatic or body characteristics. Male and female have an equal number of autosomes. And allosomes, the chromosomes responsible for determining the sex of an individual. They are called as sex chromosomes or heterochromosomes. Hetero means there are different types. The 2 types, namely X chromosomes and Y chromosomes. In human male have X chromosome and Y chromosome, but the human female have 2 X chromosomes. Next topic is karyotyping. A process of pairing and ordering of karyotype is known as karyotyping of chromosomes. Karyotype is the number, size and shape of the chromosome. A number of chromosomes in any living organism is constant. For example, in human being, each cell normally contains 23 pair of chromosomes. That is, 22 pairs is called autosomes and the 23rd pair, that is last pair, is called as allosomes or sex chromosomes. And in autosomes, chromosomes generally occurs in the pair is called as diploid. That is, autosomes, the generally is diploid. Diploid means just 2 chromosomes, 1 chromosome from father and 1 chromosome from mother, that is paternal and maternal chromosomes. In allosomes or sex chromosomes, the gamete contain a single set of chromosomes is called as haploid in number. Next, ideogram is the diagrammatic representation of karyotype of a species. It consists of the metaphasic chromosomes arranged in the homologous pair according to decreasing length, thickness, position, centromere shape, etc. And the sex chromosomes placed at the end. And next topic is DNA. So what is DNA? It is a hereditary material as it contains the genetic information. It is a hereditary material and contains the genetic information. Since it is a large molecule consisting of millions of nucleotides, hence it is called as polynucleotide. So DNA la pathina million of nucleotides irukku, that's why it's called as polynucleotide. Next, structure of DNA. It is a double helical structure. So discover James Watson and Francis Crick. James Watson and Francis Crick on the basis of X-ray diffraction studies obtained by Rosalind Franklin and Mayurick Wilkins. Watson and Crick, that is and Wilkins, were awarded Nobel Prize for Medicine in the year 1962 for his discovery of structure of DNA. And next, chemical composition of DNA. Each nucleotide consists of 3 components, that is sugar molecules, that is deoxyribose sugar, and second is a nitrogen base. There are 2 types of nitrogen base in DNA, that is purine and pyrimidine. Purine, it consists of adenine and guanine. The adenine and guanine are nitrogen base, and like that pyrimidine is consists of cytosine and thymine. So the cytosine and thymine also a nitrogen base. And third one is phosphate molecule or phosphate group. Nucleoside and nucleotide. Nucleoside means nitrogen base plus sugar is called nucleoside, and nucleotide means nucleoside plus phosphate molecule is called nucleotide. The nucleotide are formed according to the purine and pyrimidines present in them. And next, Watson and Crick model of DNA. The DNA molecule consists of 2 polynucleotide chain. 2 polynucleotide chain. 2 polynucleotide chain irukum. The chains are double helix structure and the 2 ran anti-parallel to each other, that is one another. And nitrogen bases are linked with sugar phosphate unit to form a backbone of the DNA. The pairing between the nitrogen base is very specific, that is the purine and pyrimidine are linked with hydrogen bonds. The adenine A is always linked with thymine T with 2 hydrogen bonds, and the cystone C linked with guanine with 3 hydrogen bond. Cytosine C is linked with guanine with 3 hydrogen bond. This is called complementary base pairing. Hydrogen bond make the DNA molecule stable, and each turn of double helix, that is 34 Armstrong. There are 10 base pair in a complete one turn. The nucleotide is a helix or joined together by the phosphodiester bond. And next, DNA replication. DNA replication is one of the basic process that contains within a cell. The DNA molecule produce exact copy of its own structure during the replication process. And the 2 strands of DNA molecules have complementary base pair. The nucleotide of each strand provide the information needed to produce its new strand. The 2 resulting daughter cells contain exactly the same genetic information as the parent cell. The DNA replication involves the following steps. First step is origin of replication. It is the specific point of the DNA where the replication begins. The process is The two strands open and separate at the point, the forming of the replication fork. And second step is unwinding of DNA molecules. The enzymes called helicase bind the origin of replication site. The helicase separate the two strands of the DNA. The enzyme topoisomerase, very important one, topoisomerase separate the double helix above the replication fork, and it also remove the twist formed during the unwinding process. Each of the separated DNA strand function as a template. And the third step, the formation of RNA primer and next enzyme. An RNA primer is a short segment of RNA nucleotide. The primer is synthesized by the DNA templates close to the origin of replication site. And last topic, synthesis of new complementary strand from the parent strand. The nucleotides are added with the help of enzymes DNA polymerase. A new complementary strands of the DNA is formed from each of the parent strand. The synthesis is unidirectional and leading strand. The daughter strand synthesized as a continuous, it is called as leading strand, and lagging strand, it is the other strand. Short segment of the DNA are synthesized. The short segment of the DNA is called as Okazaki fragment. The fragments are joined together by the enzyme is DNA ligase. And the final stage is termination. The replication stop when the replication fork meet at the site is called terminus, and it is situated opposite to the origin of replication site. And significance of the DNA: transmit the hereditary information from one generation to the next generation. It contains information required for the formation of proteins, and it controls the development process of life activities of an organism. Thank you very much.